Genetics is a majorly hot topic in biology right now.
Yeah, everything is genome sequencing this, gene expression that, but how much do we really know?
Well, we know a lot about which genes are present in which organisms and what certain genes do, but not a lot about how or why they do it.
Our guest today, Dr. Robert Phillips, is a researcher at CalTech who is working to understand the language of gene regulation and the methods we use to understand how genes work. This is RadioBio.
Don't know much biology.
Hello and welcome to RadioBio. I'm Lily Pennington.
And I'm Jeff Lauder. Today we're joined by Dr. Robert Phillips, the Fred and Nancy Morris Professor of Biophysics, Biology, and Physics at CalTech. Thanks so much for being with us today.
Thanks for having me. It's really a pleasure.
So could you start by telling us a little bit about yourself, like who you are and what you study?
So, who am I? I have two pictures of me surfing in my office at CalTech, and the reason they're there is not to show off, but to remind myself of who I actually feel like I really am. And so when I'm on a surf trip somewhere, like in Alaska or the Maldives, and there's no one around, I'm out in the ocean, that's in some ways when I feel most authentic. That may sound odd. So anyway, I'm a guy who grew up in San Diego as a surfer and came to science very late in the sense that I didn't like it in school, thought it was a dogmatic subject where you had to learn factoids and then regurgitate them and that kind of thing. And so I spent 7 years learning about science kind of on my own. And so it became very of deep passion, I would say, something that's like one of the main passions of my life without any shade of doubt. And then I'm also a very lucky guy, like lucky in the sense that so many times people have taken a chance on me and I've just barely navigated sort of the Scylla and Charybdis and survived. And at each stage I've just had great good fortune. So I guess I would say I have maybe 3 or 4 specific things that I'm after, but I think that they're all under an overarching sort of umbrella. And that umbrella is the question of whether or not we can have a biological science that's predictive. And when I say that, I mean it in the sense of mathematics. And the reason that I'm intrigued by that is that in this era, data in biology has become very quantitative. So I'm really excited about how physics meets biology. Mm-hmm. And I feel like biology is a huge gift to the subject of physics also. So that's the overarching umbrella. The specific things that I guess I've been most excited about are how do viruses work? So I've spent a lot of time on that. That's about to end, unfortunately. And I've been interested in mechanosensation. So it turns out a lot of bacteria actually are subjected to osmotic shocks. Lots of organisms are subjected to osmotic shocks. Even a salmon, when it goes from the ocean to the river, there's a change in the salt content of the water. And so, it's easy to take that for granted and not realize that there's some fancy machinery that allows you to go from salt water to fresh water. Mm-hmm. So mechanosensation is a big theme. And then the regulatory genome. How are cells wired up so that they can turn genes on and off in ways that sort of make sense? And then I would say I've also loved teaching evolution, and I've led like 15 field trips at this point to the Galapagos, Indonesia, New Zealand, Alaska. I love taking students out into nature, and the reason for that is that I feel like biology is fundamentally a natural science. Mm-hmm. It's about the world out there, not the world of PCR tubes in a lab.
Mm-hmm. You touched a little bit on how modern biology is more quantitative.
Yeah.
So you kind of take like an information theory approach to biology. So could you maybe walk us through a little bit about like what information theory is and how you apply it to biology, or how you bring physics into biology?
Yeah. So first of all, let me say that information theory is one of many threads that I guess that we're interested in, where you can use sort of mathematical reasoning in order to hopefully learn about how a biological system works. In this case in particular, I love the information theory mindset for the following reason. When we're young, we're taught about what's called syllogistic reasoning, where A implies B and then B implies C, and then we can make definitive statements as long as the truth of the propositions, if they're true, then we know that the conclusions are correct because they follow logically. Mm-hmm. But most of the reasoning that we do in our real lives, and even in science, is not of that character. Most of the time we have incomplete information. We don't know all the facts, and yet we still have to make a call. We have to decide what's going on. So this guy, Claude Shannon, he figured out a formal way to prove the best guess that you can make, given what you know. And so So what I would say is that this information theoretic framework is really interesting because it tells you how to, in a way, infer probability distributions about stuff like how many mRNAs are in a cell, or what are the tails of a distribution in some sort of an evolution experiment or whatever. You can make a prediction on the basis of rather meager information. So we've been using it very concretely in the context of trying to understand about the regulatory genome, because we mutagenize the DNA of the bacterium for the promoter, the particular regulatory region we're interested in. And then what we do is we measure how much we screwed up the gene, not by messing with the gene itself, but by messing with its regulation. And what we can do is we can base pair by base pair say, oh, that was a base that really mattered. And to do that, we formally use an idea that's called the mutual information. And the mutual information is a thing that tells you how variable 1 and variable 2 are tied to each other. So for example, if I were to try to choose something that's kind of where the two things are not related, like I'm going to flip a coin and I'm going to roll a dice. Those two outcomes have nothing to do with each other, so the probability of heads and 4 is just the probability of heads separately times the probability of 4. Mm-hmm. But if I were to ask about the probability of weight versus age in Americans, so there's a mutual information between age and weight. And so what I mean by that is that if you tell me age, I can win more money making bets about weight than if you didn't tell me age. You see what I mean? So that's a mutual information thing where the probabilities of two distinct things are related to each other, and knowledge of one confers knowledge of the other.
And so in your case, what are you applying this to in the cell, or in a biological system? What two things are you looking at in terms of relation to each other?
Yeah, I'm looking at whether position 35 is an A, T, or a C or G, and how much gene expression that particular gene had. So those are my two variables. That's my weight and my age.
So what does Dr. Phillips mean by position here? Well, a gene is made up of nucleotides A, T, C, and G, each in individual positions in a gene. Dr. Phillips uses information theory to determine whether a mutation at certain positions in the promoter region has an impact on what that gene does. This is similar to an experience a lot of us can relate to, figuring out what a mysterious switch does in a house. If you flip the switch and a light turns on, there is mutual information between the switch and light, but maybe you flip a switch and nothing obvious happens. Dr. Phillips, in this analogy, is kind of like an electrician modifying the switch over and over via mutation to see what it does when we turn it on and off.
So you mentioned regulatory genome. What is the regulatory genome, and why does it matter?
You know, I would say it's almost an element of a dogma at this point, the idea that we have tons of genes, no matter what organism is your favorite. You know, it doesn't matter if it's a worm or if it's a fly or if it's a fish or a bird or whatever. There's all these genes. They're not all expressed at the same level in space or time. So in an embryo, different parts of the embryo are going to have different constellations of genes that are going to be turned on or off. And then as a function of age, you know, like think about puberty, you know, or aging or whatever. Like things change over time, and there's different programs of gene expression. And so what I'm after in a way is to understand what are the rules that govern when genes get turned on and when do genes get turned off. And I'm using the words a little bit loosely because I don't really want to say on and off. It's really, there's can be a graded response, but I'm just trying to use that as an illustration. So so what we're after is I would like to be able to take any old organism, and just like I can figure out for the protein coding regions, what does the DNA sequence imply about what the protein will be, I would like to be able to look at the regulatory region and see what it implies about the rules of how the genes are turned on and off.
Mm-hmm. Could you walk us through the kind of genomic regulatory process?
We know that genomes have bunches of genes and thousands of genes. So that's awesome. And we also know from various methods that not all those genes are expressed, that the level of expression is a function of space and time. So meaning that depending on where you are within the embryo, depending on where you are in days post-fertilization or whatever, which genes are on and which genes are off, or which genes are higher and which are low, will be different. So in the hands of people like Jacob and Monod, they discovered that there are regulatory proteins whose job it is to bind to the DNA and to control when those genes get turned on and get turned off. And so the way I like to think of it is you basically have little molecular machines whose job it is to copy the DNA and turn it into mRNA, a messenger RNA. And then you have other proteins that I think of as like molecular bouncers. I don't know why I find that just to be an amusing image, but somehow I like movies and I just find it funny. Like you look at New York or LA and there's the line, and then they always show the people that arrive that are special, and they walk up and the bouncer looks at them and they open the door, and then there's all the people on the line. And so the bouncer is the transcription factor. You know, he's serving as a repressor for the people in the line and an activator for the people that come strolling up. Like I'm thinking of that show with Sarah Jessica Parker. What's that called? Sex and the City. Yeah. So there's scenes in there like that where they just stroll up. They've got their high heels on. They just walk right in, and there's all these people outside waiting and not getting anywhere. So at any rate, the upshot of that is that the regulatory parts of the genome are kind of like bouncers. They're basically controlling who gets to play and who doesn't. Mm-hmm. And so they're telling some genes, sure, you're on. It's your moment. And they're telling other genes, you got to wait. Now there's a really fun part to that, which is the external world talks to all this machinery. It says, oh, you just drank some milk. And so there's lactose around, so we got to clip the lactose molecule, or some other signal was in the environment, growth or whatever. So it's really cool to think about how small molecules are brought in from the external world, and then they impact the behavior of the molecules that are already there. That's really one of my favorite topics of protein allostery. How proteins change conformation as a result of binding to ligands and so on.
So Dr. Phillips is trying to understand what each nucleotide in a gene does to that gene's expression. The bouncer in his analogy is a binary event. The bouncer will either let you go in or not. On top of that, each bouncer has a different way of opening the door. But if there's a mutation at, for example, position 35 of the promoter region of a gene, and thus something is wrong with the door, it doesn't really matter if the bouncer is willing to let you through. But if the door is working and the bouncer lets you through, you go into the club, you dance, you drink, you produce mRNA. So Dr. Phillips studies this whole sequence of events leading to expression of a gene.
So what has been keeping us from understanding this in the past? Like, if we know how genes work, why don't we know how they're regulated?
So what I would say is that at least historically until now, or let's say until 10, 15 years ago, it was kind of a one grad student lifetime, one gene thing. Some of the new technologies like sequencing now provide an opportunity to go after these things, but it's hard. I personally am not satisfied that anybody has figured out the rules. And, I guess I feel many times the way that science works is things are confusing until they're not. I feel like we don't even know what the way forward is in some sense. And this could lead us into some philosophical debates, and it would be fun to hear your views about it because, we're living in this big data, genome-wide era, high throughput, mm-hmm. and all that stuff. And then the question is, how well will that endow us with a true understanding that's predictive? And I don't know. For the moment, I'm a little pessimistic, I guess.
And there's always the question of, is it better just to gather more data? Yeah. Or to analyze the data that we have and move on from there to see what questions stem from the data.
I find myself wondering whether science would be better off on the 5-year time scale if we had a moratorium on new data. Like for 5 years, no new data at all. That's why it's a joke. I don't really mean that, but what I am saying is that there's so much to think about. Like today, the amount of DNA that we've sequenced is the equivalent of 1.5 billion copies of Shakespeare. There's only 40 million books in the Library of Congress. So, it's like, instead of buying more books for my Kindle, I could just read the ones I already have. And so, anyway, it's a joke, but it's also, there's an element of seriousness to it, which is that the part about trying to figure out what the data means is very tricky. Poincaré, can I just say, Poincaré made this remark. He said, a science is built up of facts as a house is built up of bricks, but a mere accumulation of facts is no more a science than a pile of bricks is a house. Mm-hmm. And so I worry a lot, that we do a lot of collecting of bricks, But at some point, you've got to sit there and you've got to do the hard work of, putting the bricks together and building the house and putting a nice roof on it and cleaning the windows.
So what do? Do you analyze what we have, or are you creating more sequences?
Yeah, that's good. That's touche. Well done. I like it. Yeah. So I maybe I would say that it's also maybe sounds a little flippant, but we're trying to design methods that allow us, as we acquire that data, to actually have it teach the lesson that puts us in a position to do like the kind of mathematical modeling that I was talking about. So like the sort-seek thing that Justin Kinney did in his grad student days is really interesting, because what comes out of it is a hypothesis. So you actually have actionable hypothesis. You have a collection of putative binding sites. You have speculation about what the relevant transcription factors are. And now you can go actually do the next stage in the whole thing, which is you can try to test whether or not what you did made sense. But yeah, my group is 80% experimental, even though I'm a theorist. So in that sense, what I would say is most of the effort of the people in my lab is devoted to doing experiments, which means in the end getting data. But often they're targeted. In other words, they're designed to test something we thought we knew.
Some of your lab's work is focused on E. coli.
Yeah.
So E. coli is very familiar to a lot of people,
Yeah.
but also not very familiar. So what do we know about E. coli, and what do we not know about E. coli? And what is your research looking at in terms of filling that gap of what we don't know?
Yeah. So first of all, I guess I would say that that E. coli is it's like a celebrity in the world of biology. Like if you look at the history of modern molecular biology, what we realize is that most of the stuff that happened in the 50s and 60s was in the context of E. coli, whether it was discoveries about regulation or discoveries about the genetic code or about the triplet codons, or many, many things took place in E. coli and its viruses. So it is a scientific celebrity, but it's also very surprising, or at least it was to me when I first learned it, that for half the genes in that organism, we don't know anything about how they're regulated, nor do we have any methodology per se. I mean, you could one by one try to maybe figure out what's going on with them. So I would say that, at many levels, we do know things about E. coli and other organisms, but at other levels, we're just really beginners. And maybe that's too vague. So, concretely, we don't know how half the genes are regulated. So that's an example. Another thing that I think is really cool is that recent experiments have looked at the proteome, meaning how many proteins of each kind there are, as a function of the different growth conditions. And what was found is that depending on what the carbon source is, some things go up, some things go down, like that. I like that because there's an analogy I would make to some of the big data experiments. If I were to go to New York City and fly a drone and take a picture of yellow cabs, I could do that once, and then I could tell you, oh, look, there's the yellow cabs in New York City. But if I did it day and night, and then I did it as a function of time of day, and then we did Christmas Day and Thanksgiving, then you'd say, oh, wow, look, on Thanksgiving there's one of the streets that doesn't have any cabs, and there's these really big things going, like there's a parade or something going on, and nighttime has got a different vibe, and there's Times Square and all that stuff. It's like there needs to be context, I feel. And so I think that's in a way the thing that's really fun, and maybe that relates to your own research, because it's the ecology. How do organisms fit into the world out the window? Not that world in the lab, but how do they fit into the world, and how do they make decisions based upon what's happening to them?
Mm-hmm. Whenever I read about or hear about transcription and regulation of gene expression, I always wonder, how do you know that your methods are telling you what you think they're telling you? Yeah,
it's a great point. So I don't know if I almost could have paid you to ask that question because it's so passionately at the center of my own feelings about science. So there's many, many ways to measure gene expression. So I love your point. And I mean, you could probably guess the answer in my group, which is we do boring stuff. We do it all the different ways. We try to find out, do they agree with each other? Mm-hmm. And, this is one of the biggest moral lessons of my scientific life.
We touched on earlier, but going back to like information of biology, how are cells like circuits, and how are they not like circuits? And how does that kind of apply to our understanding of biology?
Yeah. I don't know the answer to that, to be perfectly honest, but I think that if you cut the biology down into the little tiny pieces, like one gene, if I look at one gene and under very nicely controlled circumstances, that I might be able to play the game of, oh, this is kind of looking like a circuit, like an IV curve, which is the current voltage relationship of a circuit. But if I push much harder, I start to worry that there's context dependence that we don't understand, and that could completely screw up that story. Depending on the circumstances, if I change the growth media or I change the temperature or whatever, even though I may have succeeded over in this other circumstance, I move to the next one and then I'm at sea again. It'd be like, like saying that I could predict the tides one year but not the next, or something like that, depending on if there'd been a hurricane or something like that. So the context dependence is really troubling, I guess. And that's the reason I say I don't really know, because I think the only way we can find this out is by doing case studies, the hard work of one by one, just suffering and trying to figure out, do we know what we're talking about? It's hard work. And so that's the project of a student in my group, Griffin, who's at this point trying to see, okay, if I change the growth media, or if I change the temperature, will the parameters that we think we established actually hold? So, like if you look in a data book, if you decide you're going to build a bridge and you look up the properties of steel, you just use them. It's not like, that you measure the properties of steel and then you know them.
Yeah.
But the question is, do I know the binding properties of a repressor molecule for DNA once and for all? Or if I change the growth conditions, do I have to refit them like every time? It would be like every time I build a bridge, do I have to measure steel again?
Yeah.
That would be pretty disappointing.
Specific to the conditions you're building the bridge in.
Exactly.
Yeah.
Yeah. So that's the question. Yeah.
It seems overwhelming when you think of it that way. Like, do we need to know this for every organism, for every species, for every gene? Yeah. So you mentioned in the beginning that you work with viruses, but that work is coming to an end. So what do you know now about viruses that you didn't know before? And what did your work involve?
Yeah, although that's a friendly and optimistic way to frame the question. And, part of my answer is going to be not unfriendly, but pessimistic, in other words. So I guess, first of all, to explain how I got interested in viruses, when I was a professor at Brown, before I moved to CalTech, I was working in an engineering department and thinking about mechanics. And when I say mechanics, I mean the mechanics of materials, like the metals that make the microphones here. And I heard about the possibility of using mechanics for thinking about how bacteria swim from a guy called Tom Powers. He came for an interview at Brown, and I was really intrigued by that. And then I learned about an experiment done from the group of Carlos Bustamante at UC Berkeley, in which they measured how much force it takes to put the DNA inside of a virus. And so, although I've used this analogy before, I'm just going to say it again. The way I would recommend you think about it, it's like taking 500 meters worth of the Golden Gate Bridge cable and folding it up and putting it in the back of a FedEx truck. So that's the right length scale, it's the right stiffness even. So it's a lot of work. And I don't mean work like casually, oh, that was a lot of work today. I mean it in the physics, force times distance sense. It's a lot of energy expended to put the DNA inside of the virus. So UC Berkeley had a press release, which you can still find to this day, it's like 2000 or 2001, in which Carlos had talked about the pressure inside of a virus being like that inside of a champagne bottle. And they were trying to make a big deal about it, and I was curious whether that's big or small. Like, obviously, for a bottle of champagne, if the cork shoots out and hits you in the eye, that was a lot of pressure. But if you're a virus of 50 nanometers across, it turns out it's not a very big pressure. And you can calculate why it's not such a big pressure. But I thought it was so amazingly beautiful that somebody could hold on to a piece of DNA while it was being packed into the viral capsid and measure how much force is being built up while that's happening. So it really, it led me to articulate something that I've really lived by ever since, which is what we call experiments to change your life for. And what I mean by that is, it's an experiment where when you hear about it, you actually think, oh, I should stop working on what I'm working on and work on that thing. What I wanted to get out of it was I wanted to figure out what viruses go with what hosts. Who are they infecting? Like in your mouth and my mouth. We have a bunch of bacteria, we have a bunch of viruses. But even more to the point, and then I'm going to be quiet, I think we're living in a weird era with respect to viruses. What do I mean by that? In like 1880, most people thought bacteria were pathogens. That was kind of the Pasteur and Koch thing. In other words, bacteria are pathogens. And it took us a long time to realize that no, most of them are, or a lot of them are friendly. They're part of our ecosystems. We actually depend on them for our immunity.
Our microbiome,
Yeah, exactly. So I think that's the case with viruses. Like if I say virus, it just conjures up an image of a pathogen, right? So I suspect that we're gonna, in 100 years, people are like, oh yeah, no, viruses are part of natural environments. They have a role to play. You need them. Who knows? I mean, maybe someday people will intentionally infect people with viruses. So they'll be like, oh, you're missing your viruses. So that's the thing that I was really after, was who are the viruses, who do they infect, and what are they doing? Why do you have hundreds of species of virus in your mouth? Like, what's up with that? But we failed, in some sense. I mean, my former grad student might not be happy to hear me say it that way. Let's put it this way. We failed for my purposes. I didn't feel like we learned what we were going to. Life is short. It's not a strength, I guess I would say, for me. And I don't find sequence gazing per se that exciting for me. It's not a criticism of anyone else. Just anyway, sorry, that was a long-winded answer.
No, I mean, it leads to the great question of how do you even approach that kind of problem from the start? You kind of walked us through how you approached that problem.
Yeah.
But if you're combining like physics and biology and chemistry and all these things, do you start from what you know, or do you kind of seek out methods first, or how do you start answering a really interdisciplinary problem like that?
Yeah, so I mean, I'm in the middle of doing that right now, and it's none of those answers. You could make a guess, but here's the actual answer is I try to get incredibly generous, smart people that take pity on me. And, lots of times I send my grad students to go to somebody else's lab to learn how to do something. So I'm not going to avoid a method. Like if a method seems like it's the right thing, I definitely want to bring it into the group.
What are some potential misconceptions about your research and through your teaching that you have to address?
I don't know. I can give you one little mini analogy, which is that a lot of what we do involves the use of mathematics. And so if there are people that are not comfortable using mathematics, they might have a tendency to dismiss us in the same way that I would dismiss Kazakh poetry. And so if there's no translation, so to speak, of the mathematics, and you don't speak mathematics, then it's kind of easy to say, that's some physics guys blah-blahing about my biological phenomenon, and they don't really get it, or whatever. And I'm not going to say that's a misconception for me, but at least as a teacher, I find for sure that people out there don't speak the language, and then they're quick to dismiss.
It's almost a problem in the field of science as a whole, not even a misconception with certain people. It's a language issue. Interdisciplinarity is where everything's going because we're getting so put into our own little bins that we can't even communicate across those disciplines sometimes to solve certain problems.
The lack of shared language, or the lack of understanding that, hey, not everybody's on the same page as me, means that we don't communicate with each other very well within science. So what does that mean about how we talk to the world? Mm-hmm. If we can't even talk to each other, how do we imagine we're going to talk to the world?
This episode of RadioBio was produced by Akshay Parabkari and Sunita Pertiwi and edited by Akshay Parabkari. The interviewers were Lily Pennington and Jeff Lauder, and the episode artwork was created by Jeff Lauder.
RadioBio is supported by the Quantitative and Systems Biology Graduate Group and the Graduate Division at the University of California, Merced. For more information, you can visit our website at radiobio.net or follow us on Facebook, Twitter, or Instagram.